begin, this diagram is a close-up of a muscle fiber. This segment right here of the muscle fiber is called a sarcomere, and that's what we'll be focusing on. A sarcomere is the smallest unit of skeletal muscle, and its function is to facilitate muscle contraction. The sarcomere contains filaments called actin and myosin. So here's our myosin here, the three, and then our actin are these sections here. Actin and myosin are cellular proteins. The actin and myosin, along with other components, attribute to the sliding filament theory, which essentially states that the sliding of actin past myosin generates the muscle contraction. So these parts will contract here. In order for the myosin and actin to slide past one another, they must come into contact. Myosin is the thicker filament. Located on the myosin heads are attachments for actin. Actin is the thinner filament and contains binding sites for the myosin heads. Troponin and tropomyosin are two proteins that are connected to actin and inhibit actin's ability to bind with myosin by blocking the active site. So this orange part here on the actin, that is the tropomyosin, and then these little black balls here connected to the tropomyosin are the troponin, and they basically lock it on. Tropomyosin blocks the actin's binding sites, and troponin acts as a locking mechanism, keeping the tropomyosin bound to the actin. So right here where it's connected to the tropomyosin, it actually locks it onto the actin, and that needs to be unlocked in order for the actin to actually come in contact with the myosin. When a signal reaches the neuromuscular junction, acetylcholine is released. A signal is then transmitted chemically into the muscle fiber and through the transverse tubulate system, causing the release of calcium. Calcium binds to troponin, therefore changing its shape, which unlocks the tropomyosin. Tropomyosin then reacts by moving away from the actin binding sites. So right here where it's connected to the actin, when the calcium comes in contact with these parts right here, it unlocks and the tropomyosin can move away from the parts that it's actually blocking so that the myosin and actin can come in contact.
In order for myosin to pull actin inward, the breakdown of adenosine triphosphate, also known as ATP, must occur. The reaction of hydrolysis allows ATP to break into two parts: ADP, adenosine diphosphate, and inorganic phosphate. The myosin head is now activated through ATP's connection with the myosin head. Now, a cross bridge between the actin and myosin head can form. The release of the inorganic phosphate from the cross bridge results in a greater bond. So the ATP is shown here, and it's on its way to travel to the myosin head in order to activate it. So the cross bridge attaches, rotates, and detaches. These ratchet-like series of movements causes myosin and actin to overlap, shortening and lengthening the sarcomere repeatedly. These series of movements all attribute to muscle contraction. All in all, muscle contraction is the product of thousands of sarcomeres shortening and lengthening alongside one another. Without the contracting muscles, the human body would not be able to perform daily functions such as walking, talking, and eating. Without sarcomeres, the human species would not be able to survive nor thrive. Okay, now that we've explained how everything works, we're just going to indicate where they're located. So this is the myosin, so it's the thicker part of the sarcomere. And then this is the myosin head. This is where the ATP attaches and allows it to work. And this is the actin here. It's made out of a string of beads that forms a double helix. And shown here is the little balls, so these are the troponin. So that's what causes the unlocking mechanism. And the orange string here is the tropomyosin, so that's what blocks the active sites. And this is the calcium here, so this is what allows the troponin to change its shape. ATP is shown here, so this is the energy source of energy. And lastly, this is the Z line, which attaches to the actin in order for it to maintain its structure.